So hi everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry, and in the previous session we have seen about the various kinds of gene expression in eukaryotes. In this second session we are going to see the regulation of gene in eukaryotes. So the gene regulation, it's a very important topic you need to understand because it covers basically entire transcription as well as the entire translation as well as the entire genetics. So before going to the topic, you need to be very sure about what is being taught here. Clear? Because this is kind of a complex topic which actually tries to connect all the different kinds of genetic topics which we have seen previously. Okay, and only this topic makes sense. It gives you a complete picture of what exactly happens in an eukaryotic DNA and how the various how such a huge eukaryotic DNA with huge number of genes are getting expressed and regulated in a very precise manner. Okay, now we will see the regulation of various kinds of genes in an eukaryotic cell. Okay, so it is being regulated at various levels, numerous number of levels. Okay, so it could be at the level of the chromatin structure itself, at the formation of nucleosome itself. Okay, or it could be by gene amplification. It could be by rearrangement of the genes. It could be by the presence of various kinds of regulatory elements. It could be by the transcription. So transcription can occur at certain places and it cannot occur at some other places. Okay, and sometimes transcription will occur, but again you need to express different kinds of proteins from the product of from the same mRNA. Basically, if the mRNA is polycistronic, okay, so it is done by splicing. This splicing comes under the region of RNA processing. Okay, and it could be regulated even at the level of transport of mRNA from nucleus to that of the cytoplasm. It has to enter in the cytoplasm where it can be basically translated to form protein. So this area can again be regulated. Okay, by regulating this area you can effectively inhibit the movement of this mRNA to that of the from the nucleus to go inside the cytoplasm. Okay, the next one is the degradation of mRNA, the availability of mRNA for the translation to occur. The next one, at the translation level, the translation has started and the regulation, various kinds of regulation at the translation levels. So basically it is kind of a revision of all the chapters from transcription to various different kinds of RNAs to various mechanisms by which post transcription modifications has occurred, and basically the translation as well as the post translatory modifications which has occurred. Okay, now we'll try to brief each one of them. We'll try to see each one of them now. Now the first one is at the level of chromatin structure itself. So you all know the chromatins. So the chromatins always will be in a closed state. The euchromatin is the one which is going to be the transcriptionally active region, and the heterochromatin will be seen on both the sides near the centromere as well as the telomeric ends. Okay, it will be always in a closed state. The euchromatin is the region where the actual transcription is going to occur. Okay, so this chromatin is formed. How it is being formed? It is being formed by the various kinds of nucleosomes. Okay, so these nucleosomes will form a 10 nanometer fibril, then they form a 30 nanometer fibril, and after that they get totally wounded to form some kind of wounded on a core protein to form some kind of a chromatin structure, a condensed euchromatin structure. Okay, now you need to understand this chromatin structure, you need to understand the structure of nucleosome. This is the basic unit of a chromatin structure. So what is this nucleosome? Nucleosome How it will be? You have got an octamer here, and some kind of a DNA which is surrounding this octamer. How am I saying this is an octamer? Because it has got 8 units. 1, 2, 3, 4, 5, 6, 7 and 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay. So, the rest of the 4 unit, imagine it also it's on the back side of this. Okay. Now, again this thing is surrounding this. Okay. So, each of this is called as a nucleosome. Okay. So, this strand is nothing but your DNA. Okay. This strand is your DNA. Okay. So, this DNA has been wound around some kind of a structure. Okay. These structure are called as histones. These are called as histones. Okay. Now, these histones are what charged? These histones are positively charged. Okay. These histones are what? These histones are positively charged. Because of their positive charge, they actually are able to connect with that of the DNA. DNA has got what kind of a charge? DNA has got a negative charge. So, it is through this kind of a simple charge mechanism that the DNA, the high negatively charged DNA wounds with that of the positively charged histones. Now, what about this octamer? This octamer or histones I told, right? There are different kinds of histones, different types of histones. Okay, I have discussed this in the